Okay. So hi, everyone. Thanks for coming. Happy Friday. This is our fourth Meet the PI seminar. And thank you all I see a lot of familiar names. Thanks everyone for continuing to come to these seminars. I hope they've been informative. So this is the last one of 2020. We have two more coming up in January and February. This Meet the PI is special because it is the only and first and only neuroscience themed Meet the PI event. Yay. And both Lynn and Tim are taking rotation students, right? So if anyone's interested in their work. So yeah, I would like to thank Dr. Groh and Dr. Mosca for participating in this event and being, for investing time and being so eager to join the series. And I'd like to take this time to really hear about their unique experiences and how their past experiences brought them to where they are today. And I hope this benefits anyone who is hopefully considering a career in academia. And this will give us some decent amount of advice. So yeah, thank you everyone for coming. And I think Tim is going first. So what we're gonna do is if anyone has any questions about their work specifically, you can ask after the presentation, but in terms of a Q&A discussion on career paths, we can save everything for the end and we can have one large discussion. So yeah.
Right. Well, thank you, Gina, for putting together. Thank you to the GSA, because I didn't ever have anything like this coming up where you could hear these things and kind of like when I was coming up, which admittedly was not too long ago. You did a postdoc. Mm-hmm. Anytime you posed this idea of doing something else, it's like, well, no, that's just not what we do. So I think it's great to kind of see different paths and kind of different aspects of that. I unfortunately I found out yesterday that this involves slides. So I have not put together a presentation for today. What I have a student who has an NRSA due, and I have a couple grants due. So unfortunately, those had to take precedence. But I've over the last few weeks, I've been thinking a lot about what I wanted to talk about here. And I used to have a one of those page a day calendars, a page from it posted up. It was like great quotes or something to that effect. And I'm paraphrasing this here, but it was everybody has a problem, is a problem, or is dealing with a problem. And you never know which it is. And it was one of those things that was like, okay, we should be a lot more understanding because everybody is going through something different than what other people may expect or what may be shown kind of outwardly. And intriguingly enough, it was a quote from Lucille Ball of I Love Lucy fame. So it was a very profound and deep quote, which you might not always expect from a Hollywood star. And that kind of really stuck with me. So I guess outwardly, I am cis hetero white guy. I have a lot of privilege that comes with that. And I certainly have benefited from a lot of that privilege. But I want to kind of give a couple of different aspects of this to show that, you don't have to have all of those things and still be able to make it in science and be able to do things. So I was born in upstate New York in the early 80s. Let's just leave that right where it is for timing. So I was born in upstate New York, and there is no kind way to say this. I was born into poverty. We were poor. I lived in a very, small rural town where the, expectancy of what you would do is if you got to the point where, you were driving the garbage truck for the town, or maybe working as a, Department of Transportation, work hand. That was considered to be the ultimate, and like you were living a great life. People from my town did not go to college. Maybe occasionally, every now and again, somebody would go to community college, and they certainly knew nothing about academia. My grandmother, my grandmother was an office manager for a department store, but that was later in life, mostly as a homemaker. My grandfather was a police officer. My mother was a nurse. Worked, good, steady jobs, but we didn't have a lot of money. We, aside from paying for a roof over our head, we didn't have a lot of money for food, for a lot of things. In person, I usually smile to people so that they can see like how terrible my teeth are, because we had the discussions in my family of, do you want to eat or do you want to get your teeth fixed? Like, those are the kinds of things that we struggled with, and that's where I came from. I am a first generation academic. Many people in my family, didn't go to college. My mother had an associate's degree, and she went to nursing school, and worked as a nurse. But we didn't know what college was. We didn't know what graduate school was. We didn't know all of these things. And from an early time, they kind of figured out that I was mildly smart. So was able to, get good grades in grade school. Grade school. I wasn't in grad school at 6. That would have been really awkward and just really uncomfortable. And so they, different, in a different area, actually, because our town was actually so small that we didn't have a high school. So they had to like send me out to, go elsewhere to find high school. And, I had the benefit. It was a very strange, because they sent me to like 2 towns over, which is ridiculously affluent. So yet again, throughout high school, like I was the poor kid. That was, now we have much more appropriate terms for this, like food insecure. But basically that's, back in the mid 80s and mid 90s, we were the poor kids. So, I had a lot of help from many different people and many teachers who were like, we don't care where you came from, you can do something. And that was not something that people from my town were very accustomed to hearing. So, I really took that to heart, and, I took the opportunities in high school and worked hard, was fortunate enough to get into college. And it was at about that time that I was starting to learn about science. Somebody's parent was a local college professor, and they would tell me about the things that you could do with a scientific career and a scientific degree. And when I started college, it became very clear. I was like, oh, okay, if I want to do something fancy with science, if I want to help society, you do that with a PhD, like everybody here is working on getting or has received already. And I knew that that had to be, an end goal if you wanted to do something in science. So when I went to college, I, flipped through all of the manuals and found that, there was this incredibly underused and oft ignored joint bachelor's master's program at my university. And it was one of those things that if you took the right, courses, all of them had to be cross-listed at the graduate level and do some research. Which I was just discovering was a thing. Apparently, you can do research. That you would get a bachelor's and a master's in 4 years. So I was like, okay, I think I understand this now. It's like, here's what I can do. I can do this 4-year bachelor's and master's thing. I can, that way, when I graduate college, I'll be able to go get a job at a higher pay scale because I'll have a master's and I can work there for like a decade, earn up enough money, and then be able to afford to go to grad school, maybe like when I'm 33, 34, 35. And I was very proud of myself. I was like, this is a brilliant idea. Go me. But then, when I got into, doing some lab work, like I did all work study throughout college, sent some of the money home, kept a little bit, to be able to survive in college, had incredible numbers of loans and was very fortunate enough to have scholarships. And finally, it was my junior year. Like I got all the way to junior year with this idea in my head. And one of the grad students, Ken, I will never forget this, like sat me down once in first semester junior year and just went, idiot, they pay you to go to grad school. Like he was genuinely angry that I didn't know this yet. I was like, I'm sorry, what? Could you walk me through that one more time? And he's like, you get a stipend. I'm like, what's that? I had never heard of these things. And it was at that point that I learned, oh, you get a stipend to go to grad school. I was like, well, that could be a lot more fun. So that kind of inspired me to keep going and I graduated with a bachelor's and a master's. My master's is now hanging on the wall somewhere. It's really good at collecting dust. I don't really use it for much of anything, but was able to kind of use that to adjust and kind of repitch my career. I went to grad school right after undergrad. Was able to get into, a lab that I loved the science of things. So I was always interested in the brain. As Gina mentioned, this is the all neuro session. But what, really kind of got me through it was I was always, fascinated by the cell biological aspects of it. And a lot of times, there's not as much neurobiology and cell biology talking to each other. You may, disagree with that after hearing both presentations today, because I think we're both more on the cell biological side of neuroscience. But it was always really cool to me that you could combine those. And, working on nuclear import as a graduate student, I was able to kind of unify those things. And, was able to, got my degree in 2009 and went on to do a postdoc. Was very fortunate in kind of where I went to do a postdoc and the kinds of work that, I got onto. But I want to tell this part of the story because you don't have to have like the perfect everything and just expect that, academia as a career is going to work for you. I started my postdoc in 2010. In 2012, I published 2 papers in Nature and then failed at the job market for 3 straight sessions. And I got interviews, but no, in the end, nobody, offered me positions. So, I had a K99, one of those transitionary grants, K99 to R00, did what I thought was, really groundbreaking work on synapse formation and synaptic organization and the wiring of the nervous system that we have kind of parlayed in some of the work that we do in our lab now. But, it just didn't work out. It wasn't what people were looking for. The departments, there wasn't good fits. Fortunately, in 2016, on the last possible second, the 11th hour, I was offered a position at Jefferson, and it was a great offer, and it was a great community, as everyone here knows. So I was able to kind of come to Jefferson and open up a lab. And since then, we opened the lab I started at the end of 2016. We opened the lab in October of 2017. Renovations take a while. And since then, we have been kind of studying how connections in the nervous system form. So how synapses form, how synapses mature from these kind of new incipient connections to these robust plastic entities that we know and love and read about in textbooks. And one of the projects that we've been really excited about lately is understanding how to connect, neurodevelopment, the things that happen early in life, with neurodegeneration, the things that are happening in the end of life. Typically, they are very disparate concepts, but we have found, some molecular pathways that actually suggest that there is a strong neurodevelopmental component to neurodegenerative disease, and that many neurodegenerative disease models actually show previously unappreciated defects in early neurodevelopment. So the group that I am fortunate enough to work with every day has been really kind of pushing forward, on all of those questions, and it's been a pleasure to kind of work with that. Within kind of here at Jefferson, maybe the last thing that I want to say about this is, to come back to the original point, everybody's going through something different, and everybody's going to take a different path to get through academia, to get to academia. And one of the most important things for me in running a lab is putting together an environment where it does not matter where you came from, who you are, what you think, what group you belong to, what group you identify with. Science is a place for everyone. We have the ability in science to be judged on the core of our character and by kind of the thought process that we are capable of. And that is kind of how I run the lab. And really, on our website, we have a mission statement, and kind of that ethos is very kind of encapsulated in, perhaps pithy language. But, that's the core thing that we believe in, that it really, at the end of the day, science is for everyone. Just flat. You do not need any more qualifiers to that. Regardless of who you are, what you've gone through, use that to make you a better person, use that to make you a better scientist. Anyone has a home in the kind of scientific community that we want to create. And I think that moving forward, that's one of the most important things to remember about this, is that it doesn't matter where you come from, it doesn't matter who you are. If you want to contribute to science, there is a place for you in science. And that's, an ethos that I feel very strongly. If it weren't for people and teachers that believed in that, the distinct possibility that I could be driving the garbage truck in the village of Menands right now. Well, not right now, because they don't pick up trash on Fridays. But, point taken. So that's a little kind of basis of where I've come from. I can answer, if people have scientific questions, I can answer them now, but if not, we can talk careers and different steps after Dr. Guo. So thank you.
Thank you, Dr. Mosca.
Yeah, thank you. That was great. Like the honesty, very inspiring.
Yeah. I don't know. That's kind of you to say. I don't know about inspiring, but I strive to be honest.
Okay, Lynn, are you ready to go?
Yeah, okay, let me go
Perfect on timing, so.
share my screen.
See, if I had slides, I totally would have gone over time.
Oh,
So
I hope I won't go over.
I'm capable of holding to time.
Can you see my slide now?
yes.
Okay.
Yeah, perfect.
So I have slide. And I had a hard time to name my presentation. So basically I'm gonna talk about how I went from PhD student to postdoc and to PI. So after I made the title slide, I was almost going to make the subtitle of my talk to Philadelphia story, because that's where everything happens. So I came from China after graduated from college in Beijing. So I went directly to Philadelphia, to UPenn, to start my PhD study. And Philadelphia is where I've been since then. So Right. So PhD, I was not in medical school for my PhD study, like all of you here. I actually was in the Department of Chemistry. So right here, this is the first building I saw in the United States. Like a senior student in my department picked me up from the airport, and then I saw other buildings, but I forgot about them. And then we got here, and then he told me this is the chemistry department. And this is where I spent the next 5-6 years of my research. So the chemistry department is a little bit different than the medical school. There's a lot of subdivisions in the Department of Chemistry. So, and even when we apply for grad school, we have to select a subdivision. So I think I did physical chemistry. And then when I arrived, I was a little bit kind of choosing between physical chemistry and biological chemistry. So in the end, interestingly, I did biophysical chemistry because when I was in college, I learned how to use CD to look at the protein structure, and I thought that's what I wanted to do. I want to use develop instruments to study biological problems. So that's why I did a PhD in biophysical chemistry. So in Dr. Feng Gai's lab, where he develops a lot of laser-based spectroscopy techniques to study protein folding. So there are a lot of fancy machines there, the 2D IR. He has a temperature jump machine where you could increase the temperature of a protein in like a picoseconds and then denature the protein, and then follow how it refolds. So you can see how things happen in real time really fast. And then also he develops some non-natural amino acids as a prop to see how protein folds. So unfortunately, I didn't learn any of these. So when I joined his lab, he had a homemade single molecule fluorescence microscope that is sort of set up, but not much people in the lab uses it. And then we decided to, since I am interested in using instrumentation to study biological questions, so we decided to figure out how can we use these new single molecule microscope to study biological questions. So that's what I did in his lab. It's very different from what I did for my postdoc. So we have this nice instrument, and I studied a lot of different biological systems using this single molecule technique. So we started with the unfolding of IgG, and then we started a hydration of myoglobin, and then also we were trying to understand how antimicrobial peptides kills bacteria by disrupting their membrane structure, and then we also look at influenza A proton channel protein. And then at the end of my PhD study, I started to look at the aggregation of the IAPP peptide that is aggregated in type 2 diabetes. So this is the project I never finished, but it got my interest in protein aggregation and how protein aggregate is involved in human disease. And I was really interested in understand. At that time, I was just thinking about how can we understand why does this thing aggregates? Why does the aggregate cause trouble? So that actually leads to what I wanted to do for my postdoc. So you can see that I work on so many different biological problems. But I think I'm lucky to find what I wanted to do for the rest of my scientific career at the end of my PhD. So other things I learned at grad school, my PI always told me you should start writing your thesis from day one in the lab. And this is easier said than done. And to be honest, I wrote my thesis 2 weeks before my defense. So I didn't know what he was talking about by starting writing your thesis at day one. And I only started to realize what it means until recent years. I think that means you should start thinking about your project from the very beginning of your PhD study, like what you want to accomplish and how you want to drive your project and to which direction. And then you could start writing down your thoughts, and then it will be the later years in your graduate school will be much easier. Another thing is you should always be the driver of your projects, and your publications follows you for a whole year scientific career. So that's very important, to be the owner of your own project and think about your publication. And then the last thing, I read chemistry, so of course I learned chemistry in chemistry department. But what I really wanted to say here is I actually learned these in my undergraduate. One of my undergraduate professor told me that what is chemistry? If you separate these words, it means chem is try. So you really have to try a lot. So basically you're gonna try everything you can think about, and don't be afraid of trying. So that's very important. You're going to fail a lot. And then just, think about why you fail, and start from there and try another thing. So, right. So I guess this apply to chemistry as well as biomedical research and all kind of science. So when I was finishing my PhD study, and then obviously the question is, what should I do next? Should I go to the industry or academia? And my PI back then told me, if you would get a job in the industry right after PhD, it's kind of a one-way traffic, so you could never come back to academia. But if you go do a postdoc, you would still have the choices later. And another thing is, being in the chemistry department, a lot of the industry jobs lies in like polymer companies, material science companies, like those kind of industry job that I'm not really interested. I wanted to do more biomedical. Like if it's an industry job, I would prefer a pharmaceutical company. So for me, doing a postdoc, it's like a very natural choice. So then my PI said you should change your direction a little bit. You shouldn't do exactly the same as what you did for your PhD. So I should either move more towards a physical chemistry or more towards biological chemistry. And thinking about my research interest, I decided to do a more biological relevant postdoc. And so most of my postdoc application I sent out are in the medical school. So I wanted to see how basic science can be applied to biomedical research. And then another thing I think is very important is lab environment. So both of my PhD lab and postdoc lab are very nice. We have a very collaborative and cooperative lab environment. People are helping each other, and there's never nasty competition between members, and that's not always the same. And there are labs that emphasizes on competition. And I was told that I would not survive in those labs. And I agree with that comment based on my personality. So for me, choosing a postdoc lab, I really wanted to go to a lab that is supportive and cooperative. That my case is kind of not typical compared to many other PIs, is that I stayed at my PhD institution for my postdoc. So that was I gave it a lot of thought at that time because my husband works in Philadelphia, so I wanted to stay in the area. And then I looked from New York to Baltimore. And then I went to New York for an interview, and then just that one day commute kind of makes me think that it would not work for me, because we're bench scientists. We need to stay on the bench. We can't spend 2 hours on the train every day. So I think maybe the best choice for me is to stay. And then, you know, people always say that it's not good. If you want to get a academia job later on, if you should diversify your training background. You shouldn't stay in the same institution. And so I asked a lot of advice at that time. I think what in the end is you need to diversify your scientific background. So as long as you are doing a different scientific area, that you're expanding your technology reservoir, you're learning new things, then it's okay to stay for if you have a good sound scientific reason. So for me, I already mentioned that I wanted to study protein aggregation and their implication in human diseases. So I found that James Shorter's lab at the medical school at UPenn is doing exactly the same thing that totally matches what I wanted to study. So I joined James' lab in 2012, and I moved from the chemistry department, so just 2 blocks away, to Stella Chan's building. And that's where I spent the next few years there. So James' lab studies protein aggregation in neurodegenerative diseases. So he studies a lot of like Alzheimer's disease, Parkinson's disease, so alpha-synuclein, A-beta, and how these aggregates. And when I joined the lab, I was interested in these 2 particular neurodegenerative diseases, ALS and FTD. So ALS is it's a motor neuron degenerative diseases, and FTD is a frontotemporal dementia neurodegenerative diseases that change the behavior of the patient. But these 2 diseases are actually related by protein aggregation. So a similar set of protein aggregates in these 2 diseases, including TDP43 and FAS shown here. So, and Dr. Shorter not only interested in studying how these protein aggregates, but he's also working on developing agents that can reverse the aggregation of these proteins. So for these reagents to be used as therapeutics. So that for me is it's a brand new idea, and I thought that's so excited. So and then later on, so I joined James' lab in 2012, and in 2015, there's this new idea being introduced in the field that protein aggregation might be mediated through liquid-liquid phase separation, which mediates the formation of many membranous organelle in cell. So that just made the field more fascinating for me, because now we know that all these membranous organelles in the cell that we don't know what they do before, they are functional and they have interesting properties, and they might be important for the formation of protein aggregates. So, so what I did in James' lab is we're trying to develop agents that could reverse this liquid-liquid phase separation as well as protein aggregates. So there were 2 projects that I was focusing on in his lab. So first one, we discovered that a nuclear import receptor, beta-2, could reverse the aggregation and phase separation of FAS. And then, so I finished this project at around 2016, and then started to work on, which initially as a side project for me, we started to develop RNAs that could reverse the aggregation of RNA binding protein. So yeah, I guess the lesson here is never ignore your side project. They could become a main project anytime. So things I learned during my postdoc study is that you should think broadly in your project. For example, before we before we didn't know how protein aggregates, or we still don't know how protein aggregates. But I think it's amazing people would introduce these liquid-liquid phase separation concept from physics to biology and really use this concept to explain a lot of the phenomena we saw in cell. So that tells me that you really need to think outside of the box and think every aspect of your projects. So another thing I learned is that you always need to write grants. So even as a postdoc, I probably wrote 10 fellowship applications, maybe more than 10, I don't know. And that's a very important aspect being in the academia. And then you need to go to conferences to meet people, to exchange ideas and show people what you're doing. And then moving to Jefferson, so again, my case is not typical. I wanted to stay local, and then I heard this opportunity in Jefferson, and I think it's a great place, and I think it's a good fit for me because there is a lot of science here doing research on nuclear import receptors as well as protein aggregation, and then there is a great ALS center here that could really help me with the disease aspect of my research. So I applied to this one position and I got one interview and then one offer and I accepted. So, and team interviewed me at my interview. Yeah, so.
And yet, even despite that, you still came.
So I came here and then keep working on developing agents to reverse protein aggregation. So we are now keep engineering this amazing protein cat beta 2 against the mutant fast aggregates. And we are studying how RNAs would have the ability to modulate RNA liquid-liquid phase separation. And then the third project actually connects my PhD training to my postdoc training. So during my PhD, I developed single molecule method to study how peptide assembles. So here, I'm trying to use the same single molecule method, but to study how this protein aggregates and assembles into liquids or solids in the future. So I guess here I just want to say you definitely want to, I wouldn't say definitely, but for me, I changed my research field a lot from PhD study to postdoc study. But I feel lucky because now I have my own lab. I can combine the knowledge and experience I learned in both labs and create something that I'm interested in. I think I could do. So so my lab opens in March 2019, and the first year so here is the lab when it's empty. If you go to my lab now, it's a lot dirtier and messier than it is in the picture. So that's what I spent the first year to do, basically fill up the lab and recruit people, and then wrote grants to support the lab. And then second year is the pandemic. So starting from March 2020. So that's where we are now. But the lab is full of instrument now. And also I was lucky enough to recruit some very amazing members to my lab. So Emma is the first technician that joined my lab last year, June 2019, and she helped me to set up the lab. And Neha was a master student in the lab, and she helped me to set up the cell culture room. And then we had some very hardworking rotation students that each one of them helped establish one part of the lab. So because of our lab was new, it's not like if you rotate in an established lab, you probably have a already pretty established project. But I am amazed by the rotation students, what they have achieved in such a short time during their rotation. So very grateful and thankful for them. And I have less luck in recruiting postdocs. So my first postdoc joined March this year, so 1 year after my lab started, and left a few months later because of the pandemic. And then just last week, I have a second postdoc joining. So I guess what I want to say is if you start a lab, just be patient with recruiting your people, and that could be, you could be very lucky, and things can go slow as well. But it's very important to find the right people and the right fit to the lab. It's important for both the trainees and the PI to find the right fit. And we were lucky to be funded by 2 of the development grants that I obtained as a postdoc. And then the first year we were lucky to get 2 foundation grants. And then this year we got a grant from the NIH. So the lab is supported, very grateful. So yeah, thank you. That's how I got here. And if you want any further information, here's our website. And again, rotation students are welcomed. So thank you for your attention.
Thank you for that talk.
Thank you. So I have a question, and this is for both people. Who was the most influential figure in your career, I guess, in starting your career?
For me, it was my undergrad advisor, actually. So I had done I did a summer program back in upstate New York. And it turned out that the person that I was working with there had just done a sabbatical with somebody from my home university. So I ended up in that person's lab. And he was really one of the first people to talk to me, not like I was an undergrad, but like I was a colleague. And to say, all right, well, we're going to record, from these muscles, for this. What do you think about this experiment? How would you do it? And that was fantastic. I mean, that was such a confidence builder. And, and he's very successful. So I was able to kind of follow that model and kind of always come back. I still talk to him, fairly frequently. We go out to dinner anytime we're at the same meeting. I send him, he sends me manuscripts that they're working on to review. I send him manuscripts that we're working on, unless I want them to call him for a review, and then I don't. So it's, somebody like that, like really the first person to treat me not like oh, good, you're another set of hands in the lab, but rather you're an independent thinker. Your ideas are just as good as anybody else's. Let's see what you've got, sort of thing. Like that was really the most important, and the one that gave me the confidence to be like, yeah, I could do this.
So for me, it's my PhD advisor and my postdoc advisor. They both inspired me in different aspects of science. And in addition to that, I think I really learned a lot from the postdocs in my PhD lab and my postdoc lab. Especially my postdoc lab. Like I feel they watching how they work really helped me to learn how to become an independent scientist. So to learn from your peers, I guess. Yeah.
One of my favorite things that I say to everybody is you learn something from every single person you interact with. This may be how to do something. It may also be how not to do something. So from every postdoc that I've had the fortune to work with, from every advisor I've learned things that I want to emulate and things that I don't want to emulate. And, it's just as important to learn the lessons of what you don't want to do or how not to do something, as it is to learn the lessons of how to do something or what you want to do. All of those are useful information. Negative data is informative. So, I think it's important to kind of never lose or never kind of fail to recognize a learning opportunity in that respect.
Yeah, I agree with that. You definitely learn from everyone you interact with. Yeah.
So I have a question, but if anyone else wants to jump in with a question, I'll let them ask away.
We're among friends. No topics are off limits.
I have a question.
What do you want to know about the future?
Yeah, Rob.
Yeah, I'll hop in here. Yeah, I guess this is kind of a cliche question, and like I don't know how cliche this is, but like other than funding, what would you say is like the biggest barrier to becoming like an academic scientist?
Good question.
I mean, wow. I could go all like anti-establishment right now.
I just know it's like I don't know what the rates are. This is like back when I was like deciding about whether to pursue a PhD or not. But it was like 8%, I think, of like PhDs end up becoming like faculty or something like that. It might even be lower. It might be a little higher. I'm not sure, but I know it's like pretty low.
It's certainly not fantastic. I mean, well, yeah, okay. I am going to go anti-establishment just a little bit. Lynn, I love your story. You have 100% success rate. You applied to one job. You got one interview. You got one job. I am so envious of that. I applied 12 jobs over 3.5 years across America, across Europe. And in the end, I had like 20 interviews and one job. So the pipeline is awful. And again, I'm talking from a very privileged point here. I went to fancy schools and I'm a cis white dude. If the pipeline for me is shit, then the pipeline across the board, especially with African American grad students, with underrepresented minorities, those are enormous hurdles that need to be recognized and they need to be addressed. And I think that's, if we can realize that, science is hard. We know, like, Lynn, I loved your chem is try thing there. It made me think of something about, one of those pithy things I heard is they call it research because the first time you search, you don't find anything. You have to do it again. You have to research. And we know that science is hard. Our experiments on our best projects, our experiments work 10% of the time. Why should we be making science harder by not recruiting brilliant minds, by not retaining brilliant minds? So I think that it's kind of the way that the structure of the system has come up that we are not doing enough work to retain smart scientists, dedicated scientists. Rob, I think is what I would see as one of the biggest hurdles to that. I don't have a good answer for how we would address it, but I think, one of the biggest things is acknowledging that it's an issue to start with and kind of not, on the student side, like, yeah, these are issues. Don't let these kind of institutionalized aspects convince you that you're not good enough to stay in science. Like, yeah, it's crappy and we need to be doing more work to fix this, but don't for a second believe that means you should be doing something else if you're like aces into science. Like if you, have an awakening and realize that you want to be a travel writer or, a chef, that's a completely different story. Like I think many people are going to want to be bakers after the pandemic. Who knows? And that's completely fine. But, don't let the negative historical aspects of academia convince you that, which they exist, that you're not good enough to do science, or that you should be doing something else. That was a little soapboxy, I'm sorry.
I think we all needed to hear that. Lynn, what would you say is the biggest, I guess, what did you call it? Like a
Yeah, like the biggest, like
hurdle.
I don't know, boundary or hurdle. Yeah.
Yeah.
Well, I think being a female in the female field, it's kind of there's a lot of disencouragement here. That's something you have to overcome. And then I guess in terms of science, I think other than grants, you need to find that question that really interests you, that you could really devote. You could try many times and still you could still not be disappointed and still try something. That's that area of science. To find that area of science, that's the difficult part, and I'm still looking for it.
Yeah, we definitely heard from our last all women's panel. I asked the magic question of how is it being a woman in a higher academia level, assistant professor, junior faculty. So that was, I liked how your answers kind of fed off each other a little bit.
Yeah.
Alana, you had a question?
Yeah, this is more for Lynn, although if Tim has any input, I'm welcome to hear it. As someone who's probably looking to stay in the area also, do you have any advice for like how to, for lack of better words, like do that? Because like I think there's a lot of pressure as like a postdoc and a faculty to just move wherever the science takes you, and like my family's here, so that's just not an option, and I'm not willing to compromise that. And I just so I wonder, is there anything that you think helped you be able to stay in the area?
Right. So first, when you are going for a postdoc, definitely looking for a different scientific area, so to diversify your training background. And then I think you could use it, you could think about maybe your advantage as you want to stay in the area, because when I was also concerning that I did my PhD and postdoc at the same institution, and people told me, if you're looking for a job in California, they won't even give you an interview because they see your training background. They know you want to stay at least in the East Coast. They're not even gonna invite you. But if you're looking for a position around the East Coast, that could be your advantage, because they know you have a bigger chance of staying. So that and then I have to say it is difficult comparing to people that have more flexibility to look everywhere. I don't deny that I was really scared as well, that if I would be able to stay. I don't know. I feel really lucky that it worked for me. Yeah.
If I can throw in ever so briefly, not having faced the same situation, I've crisscrossed the country multiple times now. Lynn brings up a really great point about kind of the scientific arc that your career experiences. Like if you can identify these aspects of how you're diversifying your education, your training, then that is really solid, a really solid argument for, the opportunities that you want to take. Maybe and pitch them in that fashion. Like I went to grad school in Boston. I did my postdoc in Palo Alto, like opposite ends of the nation. But I got hit all the time because I went from a Drosophila lab to a Drosophila lab. So even going to, different institutions, different labs, you're still going to get hit with something. And I kind of had to make the argument of, well, here's the background that I want, here's the background that I got, here's how I'm, taking this opportunity. And I was able to create, argue for my scientific arc. And if you can do that, then it matters a lot less where the opportunities are if they're in the same city. I mean, if you're staying in the same lab, that's probably not optimal unless you can very clearly identify how you are going to be different from your graduate career. And it's just using, those, not to go pithy with it, but, it's make those a feature of your career, not a bug. And, if you can make that argument, then there are so many more options that are available to you.
Thank you both.
Okay, any other questions? I have one more question and we're at time. So it's kind of going off of what you were just talking about, Tim, about like, and then I know Lynn mentioned the phrase expanding your scientific knowledge in terms of like PhD to postdoc, and then kind of like diversifying yourself. So I don't know, like, I guess this is towards both of you. Tim, I know you said you kind of like stayed relatively in the similar like background, but I'm sure there is some sort of challenge you had to overcome in terms of like learning new techniques or new skills. So I guess for both of you, how would you explain, I guess I'm more concerned about like the transition from, let's say, PhD student to postdoc. Like, how did you overcome that transition and those challenges of kind of being thrown into like, we all view it as like, okay, now you're like an independent scientist.
I guess if maybe I can answer the first half of that just with, I think there's two approaches and they both create equally impressive candidates at the end of the day, but they're not the same. And one is to kind of, as Lynn said, diversify your science, do one thing as a PhD student, do another thing as a, postdoc, and kind of come at this and have multiple aspects of science. That creates one type of scientist. The other, which I guess more around the realm of what I did, is kind of become the expert. And, if you're like, I was working on Drosophila cell biology at one particular synapse as a grad student. I went and did, Drosophila circuitry and cell biology at another in the central brain as a postdoc. So by the time I was done with my postdoc, like I was like the Drosophila guy. And, it creates kind of two different things. And it's one is, staying within a field, but getting deeper into it. And the other is kind of being able to combine, multiple fields into, some new element of science. And now to get to your question, is knowing, one of the first steps is kind of knowing how you want that to go, or knowing the kind of scientific journey that you want to be able to present helps you with that transition from PhD to postdoc. And this is that it's certainly worth it to take some time to think about that, because that really sets you up into, okay, if I know the things that I have to do and I can identify those opportunities, then that just eases the transition a little bit more, because it's not this complete black box of, oh God, what do I do? I'm a postdoc now, crap. It's okay, I have a list, I have things to do, there are details that are associated with this, and I can kind of follow this. And Lynn mentioned this a number of times, and it's a brilliant point to bring out. Watch postdocs. In the people that you train with, interact with them, take the lessons from them. One of the best experiences in my postdoc was kind of working with the mid to senior grad students, or the brand new postdocs by the time I was a senior postdoc, to say, don't worry about it. Yeah, I was the first person from this lab to apply for a K-99, the first person to get one. It was super opaque when I did it. Let me walk you through it and tell you what it is. It's okay. And kind of train, pass on that knowledge. So, postdocs are like, little faculty members. They love to hear themselves talk, and to talk all the time. So let them do that every now and again. Humor them, and listen to the things that they have to say, because they will give you those, great witticisms to demystify the transition a little bit.
Yeah, I totally agree. So think about what kind of scientist you want to be, and then make a plan, and you won't be so panic. And definitely talk to other postdocs in the lab, because they every postdoc had training from different labs, and that's a lot. That's like a treasure box. So yeah.
Okay, so good points. So yeah, so make a list. It won't be so overwhelming, okay? Think about it, reflect. Because yeah, just the whole idea is, I would personally like to maybe venture out a little bit and use my skills to kind of like broaden the field that I'm in. But it's kind of, it is intimidating, I feel like, but we'll see when I get there.
Another key point, anybody who's, and this is like true for life, but very much so for postdocs in science, anybody who says they have it all together and understand everything about what they're doing is lying. Postdocs are insane. Like the concept of the postdoc is nuts. There are a lot of uncertainties. Nobody knows what they're doing when they start.
Mm-hmm. Yeah, exactly.
And that holds up. Don't let anybody say, oh yeah, I know exactly what's going on. I know everything. No, they don't. There is a very high base level of uncertainty, and that's okay.
And also every postdoc is different, and even how they started and how they got their job is different. So there is not one single trajectory you could follow. You just need to make that trajectory for yourself work.
Mm-hmm.
Absolutely.
Okay, well, thank you for that. All right, I think we're out of time, but I just wanted to thank you both again for participating in your honest and open conversations, and giving us insight to us graduate students who will hopefully follow in your footsteps. But yeah, and thanks everyone for attending, and yeah, have a good weekend, everyone. Happy Friday.
Thank you guys
for having me.
Stay safe, everybody.
Stay safe too. Yep.
Bye.